Welcome everyone to this webinar, and we are talking about microplastics testings today, methods and approaches. And my name is Anssi Rajala, and I have a Master's of Science degree in chemistry from University of Jyväskylä, and I have worked now for 2 years at Measuelabs, and previously I worked in R&D of clinical LCMS. And I'm a testing expert here, and currently my scope of expertise at Measuelabs includes microplastic services, chromatography, including GC methods and LC methods, and mass spectrometry, and also I do a lot of other general chemical analysis. So a bit about Measuelabs. Measuelabs is a laboratory service company and we combine the capabilities of hundreds of laboratories into a single service that is available globally through one point of contact. For our customers, this means getting access to an exceptionally large number of analyses in one place. This makes it possible for our customers to streamline operations by letting us handle all outsourced testing your organization needs. We offer services from routine analysis of hundreds of samples to a specific compliance testing projects, and we handle everything with speed and accuracy. So today's agenda includes topics on what are microplastics, including current and upcoming regulations. Then we move on to methods and techniques for the analysis of microplastics, and then current standard methods there is available already. After that, we go through some preliminary questions provided by viewers, and also you can add questions during this presentation. And Vanni will help to pick up those questions from the chat. Okay, then to the agenda itself. So we start with what are microplastics and the current and upcoming regulations. So microplastics are synthetic polymer particles, fragments or fibers, size ranging from 1 micrometer to 2000 micrometers. Occasionally this goes down to 0.1 micrometer, especially in the new EU regulation, or up to 500 micrometers, in which case usually larger than 2000 micrometer microplastics are considered as large microplastics. Some of the most common polymer types we see in microplastics are polyethylene, polypropylene, polystyrene, and PET, and then also polyurethane, PPMA, silicone, BBT, etc. Many other types also, but those 4 I would consider are the most common ones. Microplastics are a growing concern as an environmental pollutant, and this is mainly because they are very persistent and chemically stable. Also, there's a possible health and environmental issues, but they are still a bit poorly understood. The possible health concerns include endocrine disruption, cardiovascular diseases, and metabolic disorders, and especially the human health concerns are especially related to the very small microplastics, less than 50 micrometers. And also nanoplastics. Unfortunately, nanoplastics are still something that is very complicated to study. There's at least some studies been on human blood clots that they found microplastics from those. And also there has been studies that's regarding brain tissue and, for example, testicular tissue, where microplastics have been found. And also we have tested human blood and we have found microplastics from there. Then there are the regulations. The most important one currently is the Commission regulation from EU Commission 2024/2055. This regulation only applies to the intentional microplastics, and it prohibits intentionally added micropolymer particles to 0.01 mass % in consumer products. However, it is important to point out that the regulation considers non-intentional microplastics and biodegradable plastics and water-soluble polymers to be out of scope of this regulation. Then there is Commission delegation decision from EU 2024/1441 that provides a detailed procedure for the analysis of municipal water for 20 micrometer up to 1,000 micrometer microplastics. The sampling in this case is carried out by passing 1,000 liters of water through a cascade of 4 filters. Then there's Wastewater Directive 2024/3019, which doesn't really include specific regulation, but mandates monitoring of wastewaters when seen relevant, for example, in case of agricultural related use of wastewater or sludge, and also if industrial wastewater, if it's somehow related to polymers. Then upcoming regulation or petition addition to the UCMR6 from EPA in US would also mandate monitoring microplastics in public water in the US. So there's not much regulations yet regarding microplastics, but they are coming. Then we can move on to the methods and techniques for microplastics. And usually there can be considered to be 2 main categories for techniques for analysis of microplastics. And these are microspectroscopic methods that combine microspectroscopy and vibrational spectroscopy to analyze particles, and then thermoanalytical methods that use thermal decomposition for the microplastics to quantify different polymers based on the evolved gases. And this is usually carried out by GCMS. So these microspectroscopic methods provides particle-based data, and the thermoanalytical methods provide directly concentrations of polymers in mass. And both are very robust and very good methods for identification and quantification of microplastics, but they are, due to the very large difference in the way they work, they suit a bit different needs better. So first, a bit deeper dive into the microspectroscopic methods. Microspectroscopic methods are a combination of microspectroscopy with vibrational spectroscopy. The microplastics are isolated in these methods usually to a filter from the main matrix. And in case of complex samples, usually also a digestion is carried out for the matrix. And this is basically the case for everything else than pure water samples. Most common method for the digestion is hydrogen peroxide digestion, and it's preferred because it does not greatly affect microplastics. It doesn't degrade the plastics. Other sample preparation methods include density separation, and also some cases, especially in consumer products, use of different kinds of solvents. So the main methods of types of microspectroscopic methods are µFTIR and µRaman. And µFTIR uses Fourier transformation infrared spectroscopy to identify the polymer microplastics on the filter extracted from the original sample matrix. And µRaman is a very similar method, but uses Raman scattering for the analysis instead of IR. So Raman is method where a laser is used and it hits the sample and most of it passes through, but a smaller fraction is scattered at different wavelengths, and this scattered light is called Raman scattering, is then analyzed. Raman's main good point is that it can analyze a bit smaller particles than FTIR because it uses shorter wavelengths of light, which then allows higher spatial resolution, while FTIR uses longer mid-infrared wavelengths, and its resolution is usually limited to 10 microns. Then we can a bit talk about the sampling. So usually we always, because the microplastics are in current world ubiquitous, we always recommend trying to avoid contamination, which is very easy usually. So we recommend using clean glassware for the sampling, washing hands before the sampling, and also not using polymer-based gloves if possible. Also, we recommend not using makeup and tie hair back, and also not using hair products that day when you're taking microplastic samples. And also wearing a lab coat, made of cotton, and otherwise not wearing synthetic materials. So how the process of the analysis of microplastics, in this case with the microspectroscopic methods, goes. So we have the sample container, and we can either analyze the whole sample, for example, in case of clean water samples. And usually the container is first rinsed with clean water to prevent the contamination from the container itself. And usually for the complex matrices, a reduced sample volume is used after mixing the sample well. Then the sample is pre-prepared, usually with digestion, especially if it's a complex matrix. And all the reagents used in this part are always first filtered also with microfilters. And then the sample is filtered on a microfilter, and the spectroscopic identification is carried out on the filter. And then a bit more, what are the good points of µFTIR and µRaman, and what are their less good points, limitations. µFTIR is the most common technique available in laboratories, and it's well suited for most matrices and analysis needs. It's the most commonly used for analysis. And its sample preparation is easier than other methods usually, and as the sub 10 micrometer particles are not important to keep in the analysis, it makes it much easier. Also, focal plane array detection enable a higher throughput for µFTIR, as µRaman usually works with point-by-point analysis, which takes more time to analysis. So in µFTIR's case, it's a bit like digital camera, and in µRaman case, it's more like laser that is going through each particle. Also, µFTIR gets less interference from inorganic content, pigments, and other additives, which also makes it a lot easier to work with. However, it has the limitation of lower spatial resolution and is only suitable for larger particles. Usually the cutoff is 10 micrometer. And also there's possible interference from water, but from our experience, this is usually not a large problem because the filters can be dried. Anyway, then the µRaman is most suitable for clean waters because it's a very easy matrix to work with, and also it can be used for other matrices if the small microplastics are very important for the study. For example, in case of the consumer products or food or human tissues. So Raman microspectroscopy spectroscopy's best benefit is the better spatial resolution, and it can reliably detect down to 1 micrometer particles. Also, it doesn't get much interference from the water. However, it does have the lower throughputs and more complicated sample preparation and interference from inorganic content, pigments, and additives due to the possible fluorescence that there is coming from these. Then we move on to the other type of microplastics analytical methods, the thermoanalytical methods. And this includes pyrolysis GCMS and TED GCMS. So these are a combination of thermal composition combined with analysis of the evolved gases. And virtually always GCMS is used for the analysis of the evolved gases, because it makes the robust quantification possible. For example, FTIR could be used for this analysis, but the quantification wouldn't be very good in that case. The sample preparation is a bit more varied in this case. However, also in this case, filters are often used, and especially for water samples. In case of TED GCMS, polymer content can be, in some cases, analyzed directly from the solid matrices. From case of pyrolysis GCMS, this is not commonly used, only for qualitative analysis. And there is no theoretical minimum for the particle size in case of thermoanalytical methods. However, due to the limitations of sample preparations, there is practical limits sometimes, quite often. And the decomposition of particles leads to that these methods can only provide the mass concentrations of the polymers, but will not provide particle data by default. The particle data size can be analyzed if cascade sieves is used in the sample preparation, though this is quite uncommon, I'd say. And also sometimes these methods can be combined with, for example, µFTIR, which would provide the particle size information. However, as the regulations, for example, are mostly in mass percentage, this naturally provides the mass percentage for the analysis, and also these are capable of analyzing also the nanoplastics, as there is no theoretical minimum for the particle size. So these are the good benefits of these methods. Quantification for these methods always requires calibration for each polymer, and also due to this, only a limited number of polymers can be analyzed, and also these methods are quite laborious for small analysis batches. Then what are the benefits and limitations of pyrolysis GCMS and TDE GCMS? So pyrolyzed GCMS is much more commonly available in laboratories and well suited for a wide variety of relatively homogeneous matrices. It provides a robust identification and quantification and result in mass per type of plastic natively. Compared to the TED GCMS, it has a better sensitivity and lower detection limit. However, the major downside is that only very small amount of solids can be pyrolyzed at once, and usually only the micro and nanoplastics are then extracted from the sample matrix. Usually the lower limit for the particle sizes is around 0.1 micrometer or 100 nanometers, and the detection limit is usually down to a nanogram, some nanograms per plastic. TED GCMS, in the other hand, is quite rare analysis technique and not very commonly available in commercial laboratories. But there is some upsides. For example, it's well suited for relatively heterogeneous samples and solid materials. And it can often analyze polymers directly from solid matrices with relatively high sample quantity. It can use tens of milligrams of sample at once, at least. And it provides similar results as in my mass per plastic type. However, it has the lower sensitivity compared to the pyrolyzed GCMS, down to micrograms per plastic type. And usually also in this case, the typical detection limit can be considered to be something like 100 nanometers. For both methods, the common downsides are the limited quantifiable plastic types and no particle size information. Though basically it is possible to, with a reference standard, to develop a method for either of these for a special polymers also. But in typical commercial laboratory, they have a list of certain polymers that are already in their catalog. So then we move on to the standards, and there is at least 4 current method standards available. There's also other related standards that we talk a bit about during this also. So first and foremost, there is ISO 16094-2, that is vibrational spectroscopy methods for waters with low content of suspended solids, such as drinking water. And this is an international standard method, and it's specifically meant for analysis of very pure water samples that doesn't require any other sample preparation steps except the filtration. There's also part 3 for this same standard incoming, that is for the thermoanalytical methods, but it's not yet released. This was just released last month, this standard. Then there's ISO 24187 that is released in 2003. It's actually a very general document that outlines the main principles for the analysis of microplastics and is applicable to all common analysis techniques and one variety of matrices. Additionally, this document provides a weight categories based on the size of microplastics, which helps to approximate the weight based on the results from microspectroscopic data. And this is also something that we've used quite a lot. Then there's ASTM D841-24, which is an American standard method specifically for pyrolyzed GCMS. And it is applicable for a variety of water samples from low to high content of suspended solids. And well, this document does not really provide a sample preparation, but it can be combined for standard ASTM D8333-20, which then again does describe a sample preparation for microplastic samples for similar matrices. Then lastly, there's also ISO 4484-2 that is dedicated for the microplastics from textile sources, and the samples in this case can be aqueous suspensions, powdery solids, or air emissions, all coming from textile sources in this case. The techniques applied here are mainly microscopy-based methods, including the µRaman, µFTIR, and also visual microscopy. And this standard can be also combined with ISO 4484-1, that is a determination of material loss from fabrics during washing. And this will basically determine how much fibers there is lost from the textiles when they are washed in a washing machine. And then the wastewaters incoming from this determination can be then analyzed with these methods to determine which kind of microparticles there is incoming into the wastewater. Okay, and then a small overview of our microplastic services. And we are now starting a campaign after this webinar for microplastics in clean water with µFTIR or µRaman, now for 190 euros per sample. In addition to this, we also provide services with pyrolysis GCMS for clean water, wastewater, soil, sludge, and also from biological samples, including human tissue samples. Then we also provide these same methods for, we also provide for the same matrices services with µFTIR and µRaman, and we are basically able to provide all of these methods for all of these matrices. In addition to this, we are also providing, in collaboration with Plastics, a microplastics-free seal for consumer products. And please contact us if you are interested to hear more about this. And then we move on to questions. We had some questions provided by the viewers before the start of this webinar, and we could go first through those and then go through some questions that have been provided now during the webinar itself. So let's get back to the European Commission guidelines for compliance with the SPM regulation EU 2023/2055. So in our view, the main approach to this regulation should be that the intent should be to prove that the products are not in the scope of the regulation. So one should provide the information that the product does not contain intentionally any microplastics that are not biodegradable or water soluble. And if there is polymers in the consumer product, then the polymers should be tested for water solubility and biodegradability. And this is the way that this should be first and foremost approached with. And there is, in the appendix 15 and 16, there is described the methods and the scope for the water solubility and biodegradability in regard of this regulation. Then there was questions regarding the process time, and this quite highly depends on the analysis. So as before mentioned, FTIR has a quite good throughput and does not need excessive sample preparation usually. So though some complex methods might need several days for digestion procedures, for example, in case of human samples. Raman does take by itself more time to analyze and also for the data analysis, so its process time is a bit longer usually. Pyrolysis GCMS takes quite a lot of work to set up, but once the method is running, it can push through quite a lot of samples at once. But for smaller sample batches, it's does not have very good process time, but for a higher number of samples, it does have quite good process time. And then can we analyze the size distribution and the chemical composition of the microplastics particles? And yes, this is possible with the microspectroscopic methods. And then do we have any valid methods of measuring microplastics in blood? And yes, we are able to provide analysis of microplastics in blood samples with basically any common analysis technique. Please note that the shipment of this kind of samples require packing with dry ice, and we recommended always using a glass vacuum trainer with sodium citrate with a laboratory blank sample included with ultra pure or microfiltered water taken into the tube with the same sampling equipment that is used otherwise for the sampling. Also, this needs extra documentations for basically to provide the information for the customs and the laboratory that the samples are from willing individuals. Then the comparability of FTIR and Raman results. So this is actually a bit complicated topic, but a vibration that is strong in the FTIR is often weak or often quite weak in the Raman or even absent, and then also vice versa. So depending a bit on the plastic, some have like better signal in FTIR and some might be better in Raman. So yeah, and also there's the difference in the detector type, as FTIR usually uses nowadays the FPA detectors and Raman relies on the point-by-point measurement. So they are comparable and should give quite similar results, but if there's a small particle or like fiber-like particle that has a weak vibration in the method, then it might be not well visible in the one that is weak in, if this makes any sense. Then also someone asked about the comparison of TGA GCMS with pyrolysis GCMS, and actually TGA is quite similar to the TED in principle. However, in TED, the evolved gases, basically the TED starts with a TGA, but the evolved gases then are captured and concentrated into a solid phase absorbent and then thermally released from that absorbent to the GCMS. And this method makes it easier to do the quantification than in DGA. So DGA GCMS is not usually used for quantification and only for qualitative analysis in our experience. Then there was also a question about nanoplastics. So the nanoplastics are basically analyzed in the thermoanalytical methods, but because the results are in mass and very small plastics have very small mass. So if there is any larger particles, in that case, they are much more representative in the result than the nanoplastics. There has to be quite a huge amount of nanoplastics to be basically in the result of pyrolysis GCMS, for example, but they are included in the results. But overall, nanoplastics is still a thing that is very, very challenging still and very much under research and development. And there is not very much standardized methods for their analysis. So this was the questions that were provided to me beforehand, and then we can move on to the questions asked during this meeting.
Yes, we have many good questions, especially Dora and Pavelko have been very active in the chat, which is great to see. I will hopefully you have time to answer all of them, but let's see. Andre starts with a question about where we are doing the analysis. I'll read the question out loud. Where do the analysis take place? In my understanding, you use partner laboratories for microplastic analysis. Do you then use different laboratories or only one?
Yes, we do use different laboratories because different laboratories have different techniques and are specialized for different kinds of matrices. We have a couple of laboratories in EU, a couple of laboratories in US, and also one in Asia that we work with.
Yes. Then Danielle is asking about tissue and blood samples, and is there some pre-sampling step or prior steps before pyrolysis GCMS?
Yes, there is quite similar method that is used for the other matrices. So they are digested with a hydrogen peroxide beforehand, but they are usually then filtered, or how do I say it? They are then concentrated and filtered with a ultrafiltration.
Yes.
So, yes.
Then this might have been answered, but I'll take it anyway. Dora is asking, can the µFTIR and µRaman methods also provide results of mass concentration, regardless of particle size?
To some extent, yes, but only in approximated way. So we can use the ISO. I have to check what was the number exactly. ISO 24187 to approximate the mass results for the particles. Of course, it doesn't give the results in mass in native way, but usually it's not that important to get the exact number in that case. It's more important to get a general idea, and usually this is like this method gives basically the worst case scenario in that case.
Good. Then a slightly more to the regulative side question from Pavelkova. Do you negotiate with ECHA to be, sorry, I need to read this myself through before I read it out loud to be able to read it correctly. Do you negotiate with ECHA to make these methods incorporated in the compendium of analytical methods recommended for the verification of compliance of REACH Annex 17 restrictions? I can answer already that we don't explicitly negotiate with ECHA, but Anssi, do you have other things to note on this topic?
Unfortunately, I don't have an answer for this. At least we haven't been in contact with ECHA regarding those.
Yes.
Though some of our partner laboratories have been part of the standardization procedures of microplastics analysis.
Yes. Then we had, I think, a couple persons asked about this. Are the methods accredited, or is accreditation for these methods being sought after?
We have some methods accredited. For example, we do have the ISO 16094-2 accredited for clean water samples. And also we have ISO 24187 accredited for water samples that have high particle load. So for example, environmental water samples or wastewater samples and such. So we have some methods accredited, and we are trying to get more methods accredited, but they are mostly still in process.
Yes, it is a very, very much developing field.
Yes.
Still.
I would like to still point out that we mostly do this analysis still in the accredited laboratories, even though the methods itself are not all accredited.
Yes, excellent addition. Then there's a question, are these presented methods available regardless how big the molecular weight of the polymers is? For example, microplastics with molecular weight 100,000 daltons?
I don't really think that the molecular weight itself does not affect much. In this case, it's more of the particle size in this case that affects the results. So the molecular weight of one microparticle, usually if it's even in micrometers, it's still several different polymer molecules usually. So the molecular size is not a thing that affects the limitations or detection limitations much. It's the particle size that is the limiting factor usually.
Yes. Then we have a very interesting question from Kimani asking about what are the main challenges that we are currently facing with the current microplastics certified reference materials on the market?
This is actually something that would be very interesting to look into, but I don't have a direct answer for this. I think I have to discuss with a colleague of mine regarding this thing.
Yes. So we can get back to the topic if you want to discuss this in more detail in the future. Then we have a question from Dora about sample volumes, asking what volume of wastewater or mass of sludges do you need to perform the analysis?
For wastewater, I would say it depends a bit on the type of wastewater. If it's a cleaner one, I would recommend sending up to 1 liter, but usually I would say that 500 liter, not liter, milliliters. 500 milliliters is usually enough. And usually this kind of analysis carried out with a 100 milliliter subsample. And was it sludge also?
Yes.
I would say that in case of sludge, something like 100 to 200 grams should be suitable amount.
Great. Well, actually, this second question is very much related, continuing on the same topic. Marta is asking about the minimum volume of biosamples, so blood and other human or animal samples.
Usually we would say that in case of fluid samples, at least 2 milliliters should be provided. I think the absolute minimum that we would basically need is 1 milliliter. So it would be best to have like twice that at least. Then for the tissue samples, pretty much the same. It's very much minimum of 1 gram, and at least 2 grams would be best to be delivered.
Yes, so quite a difference between the environmental ones and the biological samples. Then we have a question about the uncertainty of the methods. Do you have, Anssi, to share directly what the estimated uncertainty of the methods are?
I think because these are not all like very well standardized methods and very custom methods also are often used, but in case of the more standardized methods and accredited methods we have available, the uncertainty is something like 20 to 25%.
Okay. Then we have the next question about soils and which would be the most suitable techniques for microplastic analysis of soil samples.
For soil samples, I would say either pyrolysis CCMS or µFTIR are both well suited methods. Also TED GCMS might be also good, but if sample preparation can be used and then the matrix is very homogeneous, then pyrolysis GCMS is also very well suited, and it has the lower detection limit, so that's why it's usually best.
Yes. Then continuing on the different types of samples, Pavelkova is asking, are methods appropriate also for samples in not water solutions, for example, samples in ethanol, isopropanol, methanol, or hydrocarbon solvents?
Yes, those should be also possible to analyze, quite rare, but that shouldn't be a problem because they can be easily filtered. There might be, if it's something that is not a very simple matrix that need digestion, then organic solvents might be a bit problematic with the hydrogen peroxide, but but I think there might be other solutions also for that.
Yes. Then Andre is asking about, more specifically about the methods. Where does the big difference in the detection limit between TED and paralysis come from?
I think it's mainly from the because the gases are trapped to the thermal adsorbent, and in TED, GC, MS, and also there's, as it's usually done directly from the matrix, there's a lot of more noise on the signal. So it's the spectrum is much less clean in that case.
Thank you. Then let's see, we have one more question about water samples. Paula is asking about for clean water samples where the concentration of microplastics is low, but the presence of inorganic matter is high. What would be the recommended pre-treatment?
Can you ask the question again? I didn't.
Yeah, sorry. So for clean water, where the concentration of microplastic is low, but the presence of inorganic matter is high, what would be the recommended pre-treatment?
Is in this case inorganic matter as in sand or, for example, salt? It depends a bit what does inorganic matter in this case mean. If it has sand or something like that, it isn't really a clean water sample at that point anymore. So anyway, if there is something like sand, then the then it should be use the density separation to get the heavier inorganic content away. If it's basically clean water with high ionic compounds, then it can be basically just filtered and then washed with water after that.
Good. Then Maria-Liisa has a question about that there is a limitation of polymer types and how to define the correct polymer types.
Well, that's a complicated question. Depends highly on the what is the scope of the study basically. But usually the common polymers that are seen in the environmental samples are usually always included in the testing, and yeah. So, but yeah, it's impossible basically to answer that which should be.
So it depends on the judgment and the assumption of what would be most interesting to study.
Yes.
Yes. Great. We have gone through almost all of the questions, and we have a couple of minutes left. There are a few questions about sampling and instructions for sampling and sample preparation and sample shipment, and what kind of instructions or help we provide for that.
We can at least provide sampling instructions on paper or PDF, and we are also able to help to source suitable sampling equipment and suitable sampling bottles, for example. We don't currently provide those usually to our customers, but I think this is something that we will be looking into in the future. And regarding the sample preparation, we are able to provide very detailed instructions for our customers if the customer wants to do the sample preparation by themselves. But usually the sample preparation is always included in the price that is provided for us, except if it's specifically done so that it's not included in that case. But it's usually only if the customer really wants to do the sample preparation by themselves.
Yes. Then I think we can take the last question, and we have actually made it through all of the questions. I'm super happy that we did get this like many questions and active discussion. This goes back to the measurement uncertainty, and Camilo is asking what techniques can be used to confirm post ISO 24187 and the FTIR to further remove any of the uncertainty. Is that possible?
Wait, I want to read that question again.
Yes, it's quite of the last messages, so it should be rather easy to spot from the chat.
Yeah, I found it. If there is basically done a FTIR analysis for the sample and further analysis is needed, then I either would go forward with µRaman if the smaller particles is still uncertain or in interest, or then go with the pyrolysis GCMS. They always give a bit different results. They are not completely comparable with each other, but they will, in that kind of case, they specifically are able to get a good combination to prove that their lack of the microplastics in the sample.
Great. Could you put the last slide to show so we can leave the contact details? It is 4 o'clock and we promised a 1-hour session, so we are now getting done with it. I will, after we have got the recording, I will share that to everybody who has been here along with the presentation slides. And if you have any more questions or you want to discuss with Anssi further, then you can take his email from there and be in touch, or reply to any of my emails. And I see Jamie has put a hand up, so I think we can give the floor to you if you are able to unmute yourself.
Sorry, I didn't mean to raise my hand, but I do have a question. My interest was in nanoplastics in water, and I think you said that the techniques were still being developed and so on. Yeah, so I wanted to know a little more about what's the horizon on that. Like what can we expect to be developed in the next few years or so on that? That seems to be most important because all the biological effects that have been found, it has to be nano size particles. Yeah.
They're due to the limitations of the microspectroscopic methods. It has been a huge challenge to do the identification and the particle size analysis at the same time for the plastics. So there is some very high-end µRaman instruments that combine AMF, atomic force microscopy with the Raman spectroscopy, that are able to go to about 100 nanometers size range. But those are still very rare, and the sample preparation for those is extremely complicated still to get a good quality data. It's also possible to do via the cascade filtration to get the idea of nanoplastics, for example, with the pyrolysis GCMS, and that's maybe something that I would currently go through with. So basically, we could have a 1 micron filter or 5 micron filter or something like that, and then analyze the nanoplastics that are smaller than that with the pyrolysis GCMS. There is still the problem in that case that there has to be quite a lot of those nanoplastics to get over the detection limits. But as I said, the pyrolysis GCMS is still quite sensitive method.
Thank you.
There are also some kind of SCM-EDX can be used, but it's more a qualitative method than quantitative method.
Thank you.
Good. Then I think we can say a thank you from our side, Anssi, and I will get back with emails and a short feedback questionnaire as well, where you can leave your thoughts and improvements, ideas, and also suggest topics for future webinars that we will be hosting.
Yes, and thank you also from my part. This was nice to teach people about microplastics and our services.